Unlike the other mechanisms of immunoglobulin diversification, which generate B cells with radically differing B cell receptors, somatic hypermutation has the potential to create a series of related B cells that differ only subtly in their specificity and affinity for antigen. This is because somatic hypermutation generally involves individual point mutations that change only a single amino acid. Immunoglobulin V region genes accumulate mutations at a rate of about 1 base pair change per 10 to the third base pairs per cell division. The mutation rates of all other somatic cell DNA are much lower, around 1 base pair change per 10 to the 10th base pairs per cell division. As each of the expressed heavy and light chain V region genes is encoded by about 360 base pairs, and about 3 out of every 4 base pair changes results in an altered amino acid, every second B cell will acquire a mutation in its receptor at each division. These mutations also affect some DNA flanking the rearranged V gene, but they generally do not extend into the C region exons. Thus, random point mutations are somehow targeted to the rearranged V genes in a B cell. The point mutations accumulate in a stepwise manner as B cell clones expand in the germinal center. Generally, a B cell will not acquire more than 1 or 2 new mutations in each generation. Mutations can affect the ability of a B cell to bind antigen and thus will affect the fate of the B cell in the germinal center. Most mutations have a negative impact on the ability of the B cell receptor to bind the original antigen. For example, some mutations will abolish receptor function altogether by introducing a stop codon that prevents proper translation. Other deleterious mutations alter framework region amino acids that are essential for correct immunoglobulin folding. And still others alter amino acids in the complementarity determining regions that are responsible for contacting antigen. These deleterious mutations are disastrous for the cells that harbor them. These cells are eliminated by apoptosis either because they can no longer make a B cell receptor or because they cannot compete with the sibling cells that bind antigen more strongly. Deleterious mutation is evidently a frequent event as germinal centers are filled with apoptotic B cells that are quickly engulfed by macrophages, resulting in tingible body macrophages, which contain dark staining nuclear debris in their cytoplasm and are a long recognized histologic feature of germinal centers. Have a great day.